cell will not absorb any light because the light will just pass right through. So that's another adaptation that can help in times of stress. So far, so good? Very excited. So in terms of light absorption, within the chloroplast, you have these stacks of membranes called the thylakoid membranes. The stacks are called singular granular, and that's where the pigments are. So all of the light absorption in the chloroplast happens at the level of these stacks of thylakoids, the granule. And the most important pigment that you know of, chlorophyll, is going to be packed in the thylakoid membrane. So there is a small problem with this figure from your textbook. Which part of the chloroplast should be green? Only the granule should be green because that's where the chlorophyll is. The solution in the stroma should not be green, and the outer membrane should not be green. So that coloring of the figure is actually deceiving because it suggests there's chlorophyll everywhere. But there isn't. Chlorophyll is only in special complexes in the granule. So these membranes, the thylakoid membranes and the granule, should be green. The solution and the outer membrane should not be green. There is no chlorophyll there. It's just poor color choice by whoever edited your textbook. Sorry? Well, we don't have any pigments in the outer membrane, and we don't have any pigments floating in the stroma, so they shouldn't be colored green. Right. Now, this figure is from a different textbook, and it does show you that only the granule are green, which is correct. And it also shows you an electron micrograph showing you that you actually do see the stacks of membranes, the granule, when you take when you prepare the slide of the chloroplast. So in the laboratory, you can isolate the thylakoids, and you can keep them going for a few hours, and you can do experiments with the thylakoids. And we will talk about one group of experiments done by Andre Gegendorf, which relates to specifically what he did to the thylakoids. So we'll get to that in a little while. All right, so we are going to talk about what happens in terms of light absorption and how that leads to electron transport and synthesis of ATP, and how the ATP and an ADPH produced will be used for formation of sugars. But before we do that, you do have to know some terminology here. So you should know the difference between the thylakoid and the stroma and the lumen. So make sure that at some point, hopefully before next class, you are clear as to these terms. The thylakoid means these membranes, these folded membranes inside the chloroplast. The solution inside the thylakoids, that space is referred to as the lumen, and the solution outside of the thylakoids is referred to as the stroma. And the thylakoid membrane separates the lumen from the stroma. So anything that's going to be passing from the lumen to the stroma or vice versa has to pass through a bilayer membrane. And all of those transport rules that you know about polar and nonpolar molecules and ions, they all apply here. Right. So typically textbooks talk about light-dependent and light-independent reactions in chloroplast. Technically, there is no such a thing as a light-independent reaction, because all the light-independent reactions rely on the products of the light-dependent reactions. So if you didn't have the light-dependent reactions, none of the light-independent reactions will happen. So yes, they are not directly requiring light, but they indirectly require light because they require the products of the light reactions. So in the light-dependent reactions, is where an ATP and an electron carrier, an ADPH, which is similar to what you saw in the mitochondria. Remember in the mitochondria, you had an AD plus and an ADH. This just has an extra phosphate, so an ADP plus and an ADPH. It's just going from the oxidized form with an ADP plus to the reduced form, an ADPH, with an electron carrier. ATP, well, you saw ATP before. Now notice the Calvin cycle will use ATP and then ADPH and will recycle NADP plus and the ADP back for the light-dependent reactions. So the Calvin cycle, which is referred to as the light-independent part, is dependent on the products of the light reactions. So you can't really have one without the other. Right. One of the other things that is kind of similar to what you did with respiration mitochondria is looking at redox reactions. So we went over what happens when you take glucose and oxidize it. In this case, you're going to take carbon dioxide and then reduce it. So you follow this reaction here, carbon dioxide plus water, and you're going to make the sugar. You're going to make more water, and you're going to make oxygen, molecular oxygen out of this. Now, there's a slight problem with this diagram, and every textbook has one, except your textbook actually does it correctly these days, because I told the editors to change that, and they actually listened for a change. So the problem is this, glucose. Glucose is not the first product of photosynthesis. The reason that all textbooks do this is because they want to show you this is the reverse of what happens in respiration, which is technically not true because the reactions are nothing like what happens in respiration. But in respiration, you oxidize glucose, and here you are reducing carbon dioxide. Except you don't make glucose, you make it 3 carbon sugar instead of glucose. So when we go through the Calvin cycle, I'm going to remind you again that the first product is not glucose. So just remember that this is not the first product. Plants do make glucose, but that's not the first product of photosynthesis. So what happens to the carbon and the carbon dioxide? Going from carbon-oxygen bonds, yes sir? So is it going to be oxidized or reduced? As you go from carbon-oxygen bonds to carbon with hydrogen bonds, for example. So when the carbon is bound to oxygen, which one is more electronegative? The oxygen. So the electrons spend most of their time around the oxygen nucleus as opposed to the carbon nucleus. When you have carbon-hydrogen bonds, they're almost equal in terms of electronegativity. So carbon and hydrogen share electrons almost equally. So going from a case where the electrons spend most of the time around the oxygen to a case where the electrons are spending equal time between the hydrogen and the carbon at least, is that oxidation or reduction? Reduction, great. So that's not a gain of full electron, but it is considered reduction because the electrons are spending more time around the carbon than they did before. When you go from the oxygen and carbon dioxide to these cases with water and the carbohydrates, is there really any change for the oxygen? So oxygen was bound to carbon, and now the oxygen is bound to carbon or hydrogen. Is there really any change for the oxygen? Not really. For the water, for the hydrogen in the water, hydrogen bound to oxygen, hydrogen bound to oxygen. Is there any change? No. Hydrogen bound to oxygen, hydrogen bound to carbon and oxygen. Yes. Yeah, so that hydrogen is oxidized or reduced. Some of you said reduced, some of you said oxidized. So hydrogen and oxygen bound together. Who is more electronegative? Oxygen. So in water, most of the time the electrons are surrounding the oxygen nucleus. But if the hydrogen is now, some of the hydrogen is bound to carbon, carbon and hydrogen are almost the same in terms of electronegativity, which means the electrons are spending equal time around the hydrogen and the carbon. So spending less time around the hydrogen in water, spending more time around the hydrogen in the sugar. The hydrogen from the water in this case is reduced because the electrons are spending more time around it. The oxygen from water, going from being bound to hydrogen to being bound to another oxygen, that's oxidized. Because when it was bound to hydrogen, the electrons spend more time around the oxygen, but if you're sharing it with another oxygen, they're spending equal time around both oxygen molecules. Yes, ma'am? So the oxygen from carbon dioxide is not necessarily reduced, because if it's bound to carbon and then bound to oxygen, bound to hydrogen or carbon, it's the same electronegativity difference. But then you're looking at the, well, hydrogen is actually technically slightly less electronegative. So going from the oxygen from CO2 going to the water is slightly more reduced, but going from here to the sugar may not be. Because if you're looking at a ketone bond, then it's still a C double bond O, so that's the same reduction level. But the oxygen from water, when you go from there to molecular oxygen, you're looking at definitely an oxidation reaction. Because with the hydrogen, the electrons spend more time around the oxygen than they do around the hydrogen. But with another oxygen atom, they spend equal time around both oxygen atoms. So each oxygen, in effect, is losing electrons. So you always have to remember that oxygen is the most electronegative of these three elements, and carbon and hydrogen are almost equal. So if the electrons are shared between oxygen and one of the other two elements, then oxygen gets the electrons around itself more often. Right. So all of that will happen when we take a look at the Calvin cycle, the PCR cycle. But before we get to look at that, we do want to take a look at what happens in the light reaction. So we will come back to the PCR cycle, but let's take a look at what happens in the light reaction. So I'm going to focus on the pigments for a little while. So everybody has heard of chlorophyll. You may not necessarily know, unless you've done your readings already, which would be great, that there are different types of chlorophyll. So there's chlorophyll A and chlorophyll B in plants and green algae, and then some of the other photosynthetic organisms have different types of chlorophyll. So there's a chlorophyll C, for example. For plants, which will be our focus for now, chlorophyll A and chlorophyll B. So this type of graph is called an absorption spectrum, and the reason it's called that is because you're looking to see how much of each wavelength of light does this pigment absorb. So when you shine different wavelengths of light, going from the purple to blue to green to yellow to orange and red, so the wavelength is increasing on the x-axis here, how much of each wavelength does this pigment absorb? And so absorption is on your y-axis, you're measuring that, that's your dependent variable, and your independent variable is the wavelength of light, that's what you're manipulating. So if you follow this dark green line, the first line here, follow the arrow here, that's for chlorophyll A, and you can see that it absorbs maximally in the purple region here, and then the absorption drops, and in the longer blue wavelengths and green, it doesn't absorb anything, and in the yellow, it doesn't absorb anything, and then in the red, it has a peak. Chlorophyll B, which is very closely related structurally to chlorophyll A, has a totally different absorption spectrum. So there's just one functional group difference between the two. I'm going to show that to you. But that one small change in one functional group means that the absorption spectrum of chlorophyll B is very different. So it peaks in much longer blue wavelengths than chlorophyll A does. And it does have a peak in the red, but it's at a shorter red wavelength than chlorophyll A. There are also these pigments called carotenoids. So you've heard of beta-carotene, I'm sure. Well, carotenoids are more than just beta-carotene. So there are two groups called carotenes and xanthophylls, and we'll talk about them in a little detail later. But carotenoids as a whole absorb in the blue and green region, and they don't absorb in the red or yellow at all, and they reflect those. So beta-carotene is orangish because it's reflecting yellow and red. So carotenoids reflect yellow and red, they absorb in blue, and they absorb a little in the green region. Now plants have all of these pigments, and we're going to talk about why they have them all. Now here's the action spectrum of photosynthesis superimposed on the absorption spectra for these pigments. So the action spectrum is when you measure the rate of a process in response to the various wavelengths of light. So your independent variable is still the wavelength